This section explains why longevity is a complex genetic trait and how that shape dictates the way you study it. Aging has no single lever. It is a complex trait, a phenotype that condenses out of hundreds of loci across a handful of pathways: proteostasis, insulin and IGF signaling, mitochondria, sirtuins, chemosensation, and dietary restriction. Each of these is modulated by diet, stress, temperature, and their interactions. The generative move of the chapter is this: you never explain such a phenotype by naming its genes. You explain it by charting the gene space, the full range of factors that can move lifespan, and then modeling how they interact. A gene identified is a coordinate, not a cause. Longevity is the integral over the whole landscape. Everything downstream, the screens, the mapping, the QTLs, is machinery for sampling that space, and never for finding the one switch. This section reads aging as a steady state that leaks. Genome integrity is best understood as a flux balance rather than a stored quantity. Every cell absorbs a roughly constant rain of lesions from ordinary metabolism, on the order of 3,008 oxo-G lesions and 55,000 single-strand breaks per cell per day. That load is held level only by continuous repair. Kaole and Sung argue that the balance tips because the repair term decays with age, while the damage term does not. Nucleotide excision repair falls by about 0.6% per year. The glycosylases and polymerase beta wane, and so unrepaired lesions integrate upward over time. The decisive proof is perturbational rather than correlational. Mutate a single repair gene, a RECU helicase, or one of the nucleotide excision factors, and a human ages in fast forward. Segmental progeria is the cleanest evidence for the DNA damage theory. It is aging as maintenance failure made Mendelian. This section explains why cell senescence is best understood as an attractor rather than a single defining event. Senescence is an attractor. 6 interlinked building blocks lock each other through feedback loops: a DNA damage response, driving cell cycle arrest, the secretory phenotype, mitochondrial dysfunction, failing autophagy, chromatin remodeling, and altered nutrient signaling. Because they hold each other in place, telomere uncapping, mitochondrial poisoning, and autophagy in place, that convergence is the thesis. Any severe stress funnels into one self-stabilizing state because the loops maintain whatever enters them. The state fills in kinetically over roughly 2 to 4 weeks as each block engages at its own rate. The chapter's move demotes cell cycle arrest from definition to mere symptom. What defines a senescent cell is the network holding it there. The proliferation it abandoned is only a downstream mark of that network, never the thing that constitutes it. This section explains why aging works as a single, upstream target rather than a list of separate diseases. Age-related diseases are not independent targets. They share upstream causes, and that factorization is the whole move. A handful of common processes: inflammaging, senescence, failing proteostasis, glycation, and mitochondrial damage run in parallel across arteries, bone, kidney, and brain, and each one feeds many diseases at once. Split these processes, the way the chapter does, into private ones that touch a single tissue and public ones that are shared. This is exactly how the TP53 gene and the RAS gene underwrite cancers everywhere, while other mutations stay tissue local. The geroscience hypothesis is the consequence. Hit a public process and you delay every disease downstream of it simultaneously, rather than chasing multimorbidity one organ at a time. Aging becomes the shared factor you intervene on, and the same deterioration governs materials too, so the definition is not even specific to life. This section explains how one enzyme family works as a single sensor that couples the cell's metabolism to its aging program. One enzyme family reads the cell's energy balance and writes it onto chromatin. Every sirtuin spends NAD+ to strip acyl marks from lysines, on histones, on p53, on the foxos, on PGC1 alpha. So its catalytic rate tracks the ratio of NAD+ to NADH and therefore tracks nutrient scarcity. The 7 mammalian sirtuins are thus one dial, coupling metabolic state to the aging-relevant transcriptional program. This is conserved down from yeast, SIR2, which consumes NAD+ to silence rDNA and extend replicative lifespan. The family fans out by compartment, nuclear, cytosolic, and mitochondrial, but shares that single move. The chapter's wager is this. Caloric restriction prolongs health span partly through this sensor, so pharmacologically pushing it should buy longevity without the starvation. This section explains why the genetics of aging forces a whole system approach rather than a single gene. 1. After decades of genome-wide association studies, only ApoE survives as a robust longevity gene, and it explains almost nothing. That negative result is the hinge of the chapter. Aging refuses to localize in any single component, so the heritability hides in the interactions between hundreds of small effect genes and in cross-level feedback. The methodological consequence follows by necessity. If the causal structure is distributed, no component-by-component study can recover it. You must survey whole systems at genome scale and integrate heterogeneous omics rather than sequence one gene at a time. Integrative genomics is therefore the only frame matched to a phenomenon whose unit of causation is the network rather than the node. This section explains why metabolic rate does not set the pace of aging in warm-blooded animals and why heat is the pivot. Metabolic rate was supposed to set the pace of aging. Burn faster, die sooner. That is the rate of living theory. In warm-blooded animals that sign inverts, the long-lived mutant mice, the Ames dwarf, and the growth hormone, receptor knockout consume more oxygen per gram, carry more brown fat, and express more UCP1, yet they age slower. The pivot is where the energy goes. OCP1 uncouples respiration from ATP synthesis, spilling the proton gradient as heat rather than work. Longevity tracks the partition of flux, whether it is coupled to ATP or dissipated as heat, rather than its raw magnitude. Thermogenesis is protective. From that, you can predict the rest. Cold extends lifespan. Warmth masks the effect, which is why mice housed at 23 degrees Celsius, well below their 30 degrees Celsius neutral zone, quietly confound every calorimetry study. This section explains why yeast is a model organism for aging research. Aging becomes countable the moment division is asymmetric. A budding yeast mother retains the damage, the extrachromosomal ribosomal DNA circles, the old nuclear pores, the protein aggregates, and buds a daughter born rejuvenated. So each mother divides only a bounded number of times, and that integer is a lifespan you tally under the microscope. This is the chapter's whole leverage. A unicellular eukaryote with around 6,000 sequenced genes, many of them human orthologous, converts a diffuse organismal process into a segregation problem with a scalar readout. That readout is either replicative lifespan, the divisions counted, or chronological lifespan, the divisions counted, or chronological lifespan, the survival time in stationary phase. The cost is scope. Anything above the single cell, the intercellular facet of mammalian aging, yeast cannot show. Conserved pathways travel. multicellularity does not. This part explains why the study of aging turns to invertebrates. Aging in a mammal is invisibly slow and genetically knotted. The whole invertebrate move is to pick a substrate whose readout collapses onto the variable you want to perturb. In C. elegans, days to maturity, transparent, forward screenable, lifespan itself becomes a directly selectable phenotype. Mutants get isolatable phenotype. That single accident wins the insulin and IGF axis, the genes age 1, DAF2, and DAF16. This is the entire compression of the chapter. The organism is an instrument that makes the target quantity legible. The pantheon then fans out, by which facet each one renders legible. Drosophila's genetic toolkit, Hydra's non-senescence, out to Turritopsis dohrnii, chosen not to screen aging, but to exhibit its absence. This section explains why the interventions testing program exists and what it is built to prove. A drug that extends lifespan in one inbred strain at one lab has proven almost nothing. The effect could be that strain's quirk, or something as mundane as that room's water. The Interventions Testing Program, the ITP, turns a discovery claim into a robustness claim. It does this by running one identical standard operating procedure at 3 independent sites: Michigan, Jackson, and Texas. The animal is the 4-way cross mouse, known as HET3, where every individual is genetically unique, yet the whole population can be regenerated from fixed parental strains. 2 orthogonalizations do the work. Genetic heterogeneity dissolves strain, specific confounds. Triplicate sites dissolve facility-specific ones. What survives both of them, replicated across sites and across a diverse genome, is a signal likely to carry into humans. Here is the point the aging interventions arc drives toward. The endpoint is reproducibility itself. Reproducibility is the result the program treats as real, and any single survival curve counts only as evidence pointing toward it. This section explains why aging research inherited from short-lived lab animals may be teaching us the wrong biology, and what phylogenetic proximity corrects. Every anti-aging result we inherited from yeast, worms, flies, and mice is drawn from lineages selected for short life, organisms with demonstrably poor defenses against aging. What you learn is the biology of that poverty, not aging as such. The translational gap, where mouse cures die in human trials, was this sampling bias made visible. The corrective is phylogenetic proximity. Study aging inside the lineage that produced humans. Primates carry a longevity quotient near 1.92, nearly twice the lifespan their body size predicts. So their slow aging machinery is the machinery humans actually inherited. This is Ostad and Fisher's move. Never estimate a trait from a population chosen against it. Choose the model by what it shares with the target. This section explains the structural bet behind xenotherapeutics, the experimental therapy of cellular senescence. A single cell state, senescence, sits upstream of dozens of age-related diseases, poisoning its neighborhood through the inflammatory secretomy it broadcasts, the senescence-associated secretory phenotype, or SASP. That convergence is the whole bet. If one lesion fans out into cardiac fibrosis, diabetes, osteoarthritis, and neurodegeneration alike, then one intervention alike, then one intervention alike, then one intervention against it should delay them all at once. Xenotherapeutics splits into 2 moves on that node. The first is to kill the senescent cells, the senolytics. The second is to silence their secretome without killing, the senomorphics. The elegance is that senescent cells earn their persistence by overexpressing pro-survival pathways, so they are addicted. From this, you can predict the rest. One drug, many diseases, with specificity borrowed from the cell's own survival dependency.